Okay, crisis averted. Sorry about that. So again, we have tight junctions, gap junctions, or there's also little specialized structures called desmosomes. So let's take a look at these structures and what they are. The tight junctions are specialized connections between two plasma membranes. They can form something called an adhesion belt, where the lining of the intestine tract are actually connected together. This is going to prevent water and solutes from getting across the cells. For instance, when that food's passing through the intestines, it's not just going to go right into our body. It has to be absorbed appropriately. So it's basically isolating what's in the lumen of the intestines from the surrounding structures. And then there's gap junctions. Gap junctions are going to allow for an exchange of materials in between cells, and it's usually ions to propagate some type of cell-to-cell communication. So these cells are connected to each other, and then there's little like holes in between them that's going to allow little messages to pass right on in between, sending signals, coordinating contractions, etc. So here's what you can kind of picture for these gap junctions. Again, little specialized holes in between the cells, allowing for that information just to come on right between, or in the next picture, you can see them a little better. These specialized holes or pores allowing for that communication. So the desmosomes give the cells a little bit of bending and twisting to allow for some flexibility between the surfaces. So these are things that would be present on like our skin cells, allowing for our skin to kind of stretch and bend a little bit. That is an example of a desmosome, or a macula adherens. It's the same thing. So again, these desmosomes are just specialized proteins in between cells, allowing them to bend and twist a little bit. When our cells attach to that basal lamina, or that basement membrane, there's two layers. There's a clear layer, which is a thin layer just secreted by the epithelium itself. It has little special proteins in it. And then there's a dense layer, which is produced by the connective tissue. So it's kind of like a peanut butter and jelly sandwich. You have your epithelial cells, and then you have your other stuff, and in between are two layers. There's that clear layer and the dense layer. The dense layer is coming from the connective tissue, the clear layer is coming from the epithelium, and together it forms a nice little cement to lock everything together. And here you can see the clear layer and the dense layer of that basal lamina. The epithelia is maintained by special germinative cells called stem cells, where they undergo mitosis, and that mitotic layer is near the basal lamina, so it's near that border with the other tissue. Epithelia is classified based on shape and based on number of layers. It can be squamous, cuboidal, or columnar. Those shapes make sense. Squamous is flat and squishy, cuboidal is like a cube, columnar is like a column, or it could be simple and stratified. So you combine these two terms together and you get stratified squamous, which is many cell layers of flat squashed cells, right? So we're going to talk about all of these. Here you see simple squamous, one cell layer of thin, simple cuboidal, one cell layer of cubes, or simple columnar, one cell layer of columns. Or stratified squamous, lots of flat cells, stratified cuboidal, lots of cuboidal cells, and then stratified columnar is a little tricky, but the idea is it's classified by that top or apical cell layer. So these cells on top are columnar, therefore this is stratified columnar epithelium. So let's talk about simple squamous. Simple squamous is just one cell layer thin, very delicate. Its job is for absorption and diffusion. It's allowing things to pass through. So this would be like an inner lining of like a blood vessel. Our heart and blood vessels have this endothelium layer of within them that is just one cell layer thick. So here you can see simple squamous. And this all, again, comes from your book, and similar images in your lab book. They all kind of complement each other. But the idea is within these special serous membranes, it's all simple squamous. It's also in your lining of the lungs, the alveoli of the lungs, to allow for gas diffusion into and out of the body. Parts of the kidneys have simple squamous as well. big idea is to start understanding the relationship between structure and function. This is a thin, one cell layer thin. So it's not there for protection, right? It's not going to really be absorbing anything because there's no space to absorb. So this is just one little boundary to let things pass through it, really is all it is. You get stratified squamous. Now we have several layers of this thin stuff, and now we can start protecting because we have lots and lots of layers there. So this is where there's going to be lots of friction. There's usually a protein called keratin associated with stratified squamous. Keratin, as we'll learn in the next chapter, is this kind of dried out, crusty protein that can overtake cells. It's what makes up our hair and skin and our fingernails. And epithelium can be keratinized or non-keratinized. And again, we'll look at the differences later, but the big idea is the keratinized epithelium is like the top layer of our skin. It's kind of dried out and crusty compared to the inner linings of like our cheeks. So stratified squamous, anywhere where there's friction, inside of our mouth, inside of the vagina, inside the anus, right? Anytime there's stuff passing over the surface, it's going to be stratified squamous. Cuboidal epithelium is usually simple, not stratified, and it looks like little boxes, as I said, and this is for absorption and secretion, mostly found in the kidneys. Stratified cuboidal is only in a certain ducts, like the mammary ducts, sweat ducts. So you see them, and you can imagine as the stuff's passing through the kidneys, it's going to be absorbed and maybe even stored in these little cuboidal cells. And there's the stratified cuboidal, very not as common, but again, secretion and absorption, similar function. There's a weird epithelium called transitional epithelium. This is not really squamous, not really cuboidal. It moves in between the two. So it tolerates stretches of, or tolerates cycles of stretching and recoiling. This would be found in a place like the bladder, where it's going to fill and those cells are going to squish, or it's going to be emptied and those cells can plump back up. So it goes through cycles, and that's why it's called transitional. So when the bladder is empty, those cells are nice and puffy, and when the bladder is full, those cells get squished down because it's full of urine. It's a transitional epithelium. Then we have columnar. Columnar epithelium can actually be classified three ways. Simple columnar, this is for absorption and secretion, found mostly in the small intestine. Pseudostratified columnar, which has cilia, it's usually called ciliated pseudostratified columnar epithelium, very big term. That's found in the lungs, portions of the upper respiratory tract. Or stratified columnar, which is very rare, portions of the respiratory tract, where again, only that superficial layer is columnar. So that simple columnar, as I mentioned, has that microvilli on it in the small intestine to increase absorption rates. And you can imagine as that food is passing over through the lumen, all of those microvilli are absorbing the nutrients. We'll talk all about that next semester, getting absorbed into these cells and then possibly entering the bloodstream. Whereas pseudostratified ciliated columnar epithelium has the cilia on it, which kind of wave back and forth, and we'll talk about the function of that later, but the general idea is trapping any dust and debris, and then, of course, allowing for the passage of materials, of air down into the lungs so that breathing can occur. The stratified columnar, again, is very rare. This is in small, isolated areas of the respiratory tract and digestive tract. So let's talk about glands. We have endocrine and exocrine glands that are epithelial tissues. The endocrine glands release hormones. We'll focus on those when we get into the last chapter of the book for this semester. Is it 17 or 18? I can't remember. And then exocrine glands are the glands we're going to talk about now. These are the ones that produce secretions which go onto the body surface. And they usually use a duct. So there's three types. There's merocrine secretion, apocrine secretion, or holocrine secretion. Merocrine secretion is when the secretion is coming out through a vesicle. So it's like an exocytosis. Whatever's leaving the cell isn't ruining the cell, it's just kind of leaving the cell through a little vesicle bubble. Apocrine secretion is where a little part of the cytoplasm comes out with the secretion as well. The mammary glands, milk, is released in this way. Holocrine secretion is where the entire cell bursts and is destroyed by whatever the secretion is. So this would be like a sebaceous gland inside of the hair follicle. When the hair follicle is moved by the erector pili muscle, a little bit of that sebaceous gland is squeezed, and those cells are literally bursting with sebum, with oil, which comes into the surface of the hair. So talking about how the epithelium is classified, there's merocrine glands, and again, here you can see that exocytosis, just the general sweating, or in this case, we're looking at a salivary gland secreting. The mammary gland, just breaking down part of the cytoplasm containing the milk, or a holocrine secretion where the entire cell just kind of explodes and erupts its secretion along with itself. There are different types of secretion. So there are different ways that things are secreted, as well as different things that are secreted. So the stuff that is secreted can be watery. That's a serous gland. It can be mucousy from like a mucus gland, or it can be mixed, which would be both. So let's talk about some of these mucus glands. They can be unicellular. In the instance of goblet cells, which are lining the digestive tract, they're just kind of scattered amongst the intestines and then help excrete mucus into the digestive tract so that food can just move through without any friction. They can also be multicellular. So they can be simple, they can be compound, they can be shaped like a tube or shaped like a pocket, and so they're classified in different ways. A simple gland is just like one little pocket, right? These are all simple glands. Tubular is just a tube. Coiled means it's coiled, or branched tubular means there's several branches, but it's still shaped like a tube. Same thing with alveolar. Alveolar is more like a pocket than a tube, and it can be branched or simple as well. We look at compound glands, and there's lots of branching that occurs. Here's a compound tubular, compound alveolar, or compound tubular alveolar, which is all of the above. Do you have to know which glands are which? No, but you should know examples of each. So you should know that mammary glands are compound alveolar. You should be able to tell me that one of these is a compound tubular alveolar. So either know that it's salivary, know that it's pancreas, know that it's respiratory. Oh, that's the end. Awesome. So this summarizes epithelium tissue for you. Our next lecture will cover connective tissue. I hope that this video helped you, and please let me know if you have any questions.